everyone. I'm delighted to be opening another Global IMMUNOTALK today. I am Elina Zuniga from UCSD, and I am today in the company of the co-organizer of the Global IMMUNOTALKs, Carla Rodlim from Yale University, and our terrific speaker today, Dr. Alexander Rudensky from Sloan Kettering. As always, before Carla introduced Dr. Rudensky, I would like to remind you very briefly about the goals we have set for all the Global IMMUNOTALKs and that we hope you help us accomplish. First, to benefit and inspire immunologists across the world in an egalitarian manner. For that, the talks are free, are recorded, and the discussions are via Twitter. We would also like to increase opportunities for scientific learning without traveling, making the talks accommodating and also environmentally friendly. As always, we want to remind you that the credit should go to the speakers that have generously agreed to share their knowledge in this seminar series. So thank you so much to all the speakers that are listed here and all the ones that have agreed to give talks after August 5th until the end of the year. So we will soon release the new poster. So with that, I let Carla introduce Dr. Rudensky.
Thank you so much, Elina. It is indeed my real pleasure to introduce Dr. Alexander Rudensky, our global immuno speaker today. Dr. Rudensky was born in Moscow and became interested in science, I understand, at a very early age. I believe he enjoyed various areas of science, from mathematics to botany, but became fascinated with chemistry. And this was so much that he went to chemistry school in the evenings during high school. I believe this provided Dr. Rudensky a solid foundation for his choice of studying biochemistry at the Second Medical School in Moscow. Now, similarly to our previous global immuno speakers, science really took Dr. Rudensky on a world tour. He was supposed to go to Germany, but I understand there might have been some bureaucracy on the way and could not get a visa. So luckily for us, he came to the US to continue his training with Dr. Charlie Janeway right here at Yale. Now, both during his PhD and as a postdoc, Dr. Rudensky focused on antigen presentation and processing and made fundamental discoveries, particularly on the identification of the sequence of peptides bound to the MHC class II molecule. Now, after his postdoc with Dr. Janeway, Dr. Rudensky started his independent laboratory at the University of Washington in Seattle. And it is there where his research veered to the development of regulatory T cells, something that I'm sure we're going to learn much more today. And it's really thanks to Dr. Rudensky's work that we have acquired a much better understanding on how regulatory T cells develop, how they are formed, and how they function to regulate various immune responses. Today, Dr. Rudensky is the chair of the immunology program at the Memorial Sloan Kettering Cancer Center, and he's also an investigator of the Howard Hughes Medical Institute. Dr. Rudensky has received numerous awards. I won't go into the long list of these awards, but I would like to mention that Dr. Rudensky is a member of the National Academy of Science and also a member of the National Academy of Medicine of the United States. Now, on a more personal note, I have always enjoyed his methodic and elegant approach to science, and very especially his molecular precision when it comes to his discoveries. And I'm sure he is going to delight us today with new findings on the biology of regulatory T cells. So Dr. Rudensky, thanks so much for accepting our invitation to present as a global immune speaker.
Thank you very much, Elina, and thank you, Carla. Thank you for inviting me. It's a pleasure and honor to be a global immuno speaker, quite a title in these times and days.
Absolutely. It's a real pleasure to have you. And I'm sure we have many people across the world listening to you. I remember from the very beginning, we have people from Russia. So I'm sure we will have, many from Russia today. Before we start, Dr. Rudensky, you have been a scientist for a while. And in this, long path, I'm sure you have acquired a lot of experience, and I'm sure you have also become wiser along the way. So before we start, we would love to learn from you, in your opinion, what are the current biggest challenges in immunology?
I think that the current biggest challenge in immunology is probably the same as in biomedical research at large, and namely fragmentation of the field in myriads of subfields, insular and isolated, as a result of predominant reductionist approach to biomedical research, and probably not only limited to biomedical research. And in the process, I think even within the same field, the field of immunology, people working in different aspects and different areas of immunology have their own language to a degree, their own tools, and there are walls that separate these different subfields in a single field, and not if we are not even talking about biomedical research at large. And I think that this is the biggest challenge. So what I would call new synthesis, where by generalizations and concepts would allow to put together pieces of this puzzle that is broken and lost in a way, separated in multiple charts. So that's my long answer to your short question.
No, that's very, very important, how we synthesized all this myriad of information. Thank you. Thank you so much. Well, we look forward to your talk. Very much look forward to your talk.
Thank you.
All right. Thank you again. So today I will start, maybe I will start by sharing my screen, I guess. That would be quite appropriate. And can you see me? You can see screen, me, and the arrow.
All that is perfect.
So it's moving. It's moving around. You can see.
Yes. Yes.
So at no surprise, I will talk today and share with you some of our work, earlier and recent work, and some work in between, related to biology of regulatory T cells, a specialized lineage of suppressive cells whose function features prominently in diverse physiological and pathological settings, reflected in this faint font in the background. At the center of biology of regulatory T cells lies a really simple regulatory network that is centered on cytokine interleukin-2. Regulatory T cells were shown by Shimon Sakaguchi in a milestone paper published in 1995 to be a subset of CD4 T cells that expresses high level of high affinity subunit of interleukin-2 receptor CD25. Regulatory T cells do not produce interleukin-2 themselves, but when effector T cells produce interleukin-2 in response to T cell receptor engagement, that results in expansion and activation of regulatory T cells, and hence suppression commences. Now, 20 years ago, Fred Ramsdell and Talal Shatila cloned a novel gene, FORCAT family member FOXP3, and shortly thereafter, work from 3 laboratories, from Fred Ramsdell laboratory, Shimon Sakaguchi's laboratory, and Jason Fontenot, at the time graduate student in my lab, was able to demonstrate that this new transcription factor, FOXP3 encoded by X chromosome, is required for regulatory T cell differentiation and function. Subsequent work from the lab, mostly utilizing reverse genetics approaches, conditional alleles of FOXP3, various reporter alleles, showed that during differentiation of regulatory T cells, expression of FOXP3 confers suppressor function and fitness to regulatory T cells, whereas precursors of regulatory T cells we can call T regulatory T cell wannabes that express FOXP3 reporter null allele, while they share some of the transcriptional features and phenotypic features with regulatory T cells, lack suppressor function. Not only FOXP3 expression is required for establishment of functionality of regulatory T cells, its continuous expression is required for the maintenance of identity and functional competence of regulatory T cells, as was demonstrated by Luke Williams now quite some time ago, graduate student at the time in the lab, when he ablated conditional FOXP3 allele in differentiated regulatory T cells and observed that this former regulatory T cells lack suppressor function and acquire pathogenic potential. FOXP3 mutations in mice and men result in fatal and systemic autoimmune and inflammatory disease. As shown on the left upper corner, this is IPEX patient that presents with this little boys are brought into clinic because they cannot eat. They develop severe enteritis, a variety of autoimmune manifestations throughout the body. That essentially 50%, half of these patients develop, and 90% of these patients develop diabetes before 3 months of age. And similarly, mice lacking FOXP3 gene develop similarly severe and fatal lesions. These are FOXP3 knockout mice generated a long time ago by Jason Fontenot. And work by Jason showed that essentially all manifestations of the disease in these mice were due to the absence of regulatory T cells and not due to cell intrinsic regulation of T cell reactivity or dependence on co-stimulation by transcription factor FOXP3. So this studies showed that regulatory T cells are required in mice and men to prevent severe autoimmune lesions when they are present early in life. Generation of mice with a conditional ablatable allele of FOXP3 where diphtheria toxin receptor, simian diphtheria toxin receptor coding sequence was inserted into the FOXP3 locus showed that regulatory T cells are continuously required for preventing severe and fatal autoimmune inflammatory disease in mice. In both SPF and germ-free mice that harbor this ablatable FOXP3 allele, in which all regulatory T cells are decorated by simian diphtheria toxin receptor, and upon administration of diphtheria toxin are eliminated overnight. These mice, adult mice subjected to ablation of regulatory T cells, die within a very short period of time with similarly widespread lymphoproliferative, myeloproliferative syndrome affecting multiple organs and tissues. And it is noticeable, what I would like also to emphasize, that a loss of regulatory T cells or their germline deficiency results in sharp augmentation in all 3 major types of inflammatory responses currently recognized in type 1, type 2, and type 3. And this little axis is the fold increase in numbers of effector T cells producing different types of pro-inflammatory cytokines. So through generation of a conditional, through generation of a reporter allele, a functional reporter allele of FOXP3 gene, Jason Fontenot was able also to demonstrate that two signals, T-cell receptor signal and interleukin-2 or other common gamma chain receptor signaling is required for induction of FOXP3 gene and therefore for differentiation of regulatory T cells. And in the work, beautiful studies performed by Chi Shea and Mike Farrar's lab and published now 12 years ago, these two groups demonstrated that the process of regulatory T cell differentiation commences in a two-step model where T cell receptor signal first results in establishment of this regulatory T cell-like transcriptional features, not quite matching expression of genes in FOXP3 expressing cells, but closely resembling. And this include also upregulation of CD25, so the cells now become competent to receive interleukin-2 signal, and upon provision of interleukin-2 can upregulate FOXP3 expression and commit to regulatory T cell lineage differentiation. So that puts together two signaling pathways that are required in this sequential arrangement to commit precursor cells to regulatory T cell lineage differentiation. So we've been interested now for a while in understanding how cues guiding regulatory T cell fate decisions converge on the FOXP3 gene locus. One can perhaps naively anticipate that, assume that the signaling pathways might converge directly on the locus, on the gene locus, and regulate FOXP3 gene expression. And with these studies that were initiated now quite a long time ago by Yi Zheng, former postdoctoral fellow in the lab, and Steve Josefovich, a graduate student in the lab, and continued on with Yong Feng, and now two current members of the lab, Joris van der Wicken and Stas Dickey, focused initially on this four cis-regulatory elements, conserved a non-coding sequence 1, 2, 3, and 4. This enhancers were identified based on their chromatin features, accessibility, particular histone modifications, and conservation, and then were examined using genetic means, namely conditional and germline ablation of regulatory elements, and all the mice that were generated harbor GFP reporter of FOXP3 gene. So this, what I would like to discuss with you first, there are two cis-regulatory elements we call CNS3 and CNS4 that are accessible in precursor cells prior to FOXP3 gene expression. These two elements were candidates for enhancers on which signals from T-cell receptor and interleukin-2 receptor converge. So let's start first with this enhancer, entronic FOXP3 gene enhancer we'll call CNS3. Joris and Yong found that this particular enhancer is poised in precursor cells. This poised state of enhancer is established early on, already in hematopoietic stem cells and preserved throughout T-cell differentiation. And this poised state is indicated by this little red circles shown here. These little red circles indicate enrichment for a permissive histone mark, monomethylated histone H3 in position K4, lysin 4. And at a more immediate precursor stage of differentiation, presence of this monomethylated mark and CNS3 enables establishment of a poised state of FOXP3 promoter, and that and in the absence of CNS3, as schematically shown here, this poised state of FOXP3 promoter is not established. So CNS4 is somewhat similar, although it's located in an intron of an upstream gene from FOXP3. CNS4 also is poised in precursor cells and its presence enables establishment of poised state at the promoter. And in the absence of CNS4, this poised state of the promoter is not observed, or it's significantly reduced. So gene targeting studies by Young, Joris, and now Young and Stas showed that both CNS3 and CNS4 promote regulatory T cell differentiation by facilitating expression of FOXP3 gene in precursors of regulatory T cells in the thymus and in peripheral CD4 T cells when they're activated in a particular manner by cross-linking T cell receptor CD28 and by provision of cytokine TGF-beta and interleukin-2. They also found that these two enhancers were dispensable once regulatory T cells differentiate and express FOXP3 gene, because they found that ablation of conditional alleles of CNS3 and CNS4 did not result in impairment in the maintenance of FOXP3 gene expression. So the studies that I'm not going to describe, because these were published by Young and his colleagues in the lab, showed that by poisoning FOXP3 promoter, CNS3 enables broadening of regulatory T cell repertoire by inclusion of precursor cells that express T cell receptors with a lower signal strength. So if we can schematically show this, and in gray, you would see T cell receptor signal strengths, imagine T cell receptor signal strengths or functional affinity that supports differentiation of conventional T cells. And in green, it's distribution of, again, imaginary distribution of signal strengths that promote regulatory T cell differentiation. And we know from work of many labs, including Chi She, Andy Caton, and Harold von Böhmer, that stronger T cell receptor signal promotes FOXP3 gene expression. So what Young found was that, in the absence of CNS3, only receptors with this increased strengths and the higher range of T cell receptor signal strengths promotes differentiation of regulatory T cells. And functional implications of this restriction of the repertoire in the absence of CNS3 in precursor cells were autoreactive T cell generation, autoantibody generation, and frank autoimmunity that resulted in impeded diversification of regulatory T cell repertoire. So in contrast to CNS3, on which T cell receptor signaling pathway converges, Young and Stas found that CNS4 facilitates interleukin-2 dependent step in differentiation of regulatory T cells, and particularly important in neonates. So this is the second step that I showed you in the model of two-step differentiation of regulatory T cells. And that's transition between CD25 positive, FOXP3 negative precursors in the thymus that can be facilitated by interleukin-2, and that would result in expression of FOXP3 gene. So what analysis of CNS4 deficient mice and their litter mates showed that shortly after birth, immediately after birth, at early stages of regulatory T cell, postnatal differentiation of regulatory T cells, there is a buildup of this precursor cells. So this plot on the left side reflects frequency of this precursor cells shown here in this faint green circle, CD25 positive, FOXP3 negative ones. And in the absence of CNS4, as opposed to litter mates with intact CNS4, there is a buildup of these precursors. Similarly, there is a paucity of the end product. So the step between CD25 positive precursor cells differentiation to FOXP3 positive regulatory T cells in the thymus is impaired. and this block is partially alleviated this time. So this can be readily modeled in vitro, as Stas was able to show. And you can see here that when precursor cells were isolated from CNS4 sufficient mice, this is CNS4 floxed mice expressing this reporter allele, and are provided with interleukin-2. In this case, it's recombinant human interleukin-2. They upregulate FOXP3 gene, whereas that doesn't happen in CNS4 deficient precursor cells, and this is just quantification. So work that I'm not about to describe for the lack of time showed that CNS4 binds STAT5. It has 2 conserved motifs for STAT5, and in fact, it's the first site within the FOXP3 locus that binds transcription factor STAT5 activated downstream of interleukin-2 receptor signaling pathway. So to sum up function of these 2 elements, we believe that CNS3 is the element on which T-cell receptor signaling converges, and its function is to lower the threshold of T-cell receptor signals that are permissive for regulatory T-cell differentiation, whereas CNS4 binds STAT5, and this is the element on which directly interleukin-2 receptor signaling converges in the second step of regulatory T-cell differentiation. And CNS4, we believe, ensures robustness of regulatory T-cell differentiation, particularly essential under suboptimal conditions during neonatal period. One can also it's tempting to speculate that similar epigenetic mechanism may operate during differentiation of other cell types in the immune system and beyond, to enable their response to under range of growth factors or morphogen concentrations. So another another enhancer of FOXP3 enhancer we call CNS2, it's intronic enhancer. Its function is actually limited to differentiated regulatory T cells. I told you that CNS4 is dispensable for the maintenance of FOXP3 gene expression in differentiated regulatory T cells, and work from Ye Zheng and Yong Feng in the lab showed that this particular intronic enhancer, its sole function is to maintain identity of regulatory T cells during division of mature regulatory T cells. And Yong also found that CNS2 allows for heritable maintenance of regulatory T cell lineage identity when interleukin-2, as we know, major cue promoting the cell fate is limiting. And another condition when CNS2 is essential is when pro-inflammatory cytokines, factors that promote alternative cell fate, namely differentiation of effector cells, pro-inflammatory effector cells, are present. So now I would like just to briefly mention that regulatory T cells differentiate not only in the thymus, they can differentiate extrathymically. And that implies ontogenic heterogeneity of regulatory T cells. This extrathymically generated regulatory T cells or peripheral regulatory T cells are reliant on a distinct enhancer in the FOXP3 gene that was shown by, again, Ye Zheng and Steve Josefovich and Rachel Neese in the lab. So this enhancer, conserved enhancer, contains binding sites for 3 transcription factors downstream of 3 major signaling pathways that have been implicated in extrathymic differentiation of regulatory T cells, binding site for SMAT, NFAT, and retinoic acid receptor. So I would just briefly summarize quite a bit of work in the lab that was enabled by the discovery of specific function of CNS1 and extrathymic differentiation of regulatory T cells, because this finding enabled us to generate 2 mice that were impeded in extrathymic differentiation of regulatory T cells and explored function of extrathymic extrathymic regenerated regulatory T cells. And the work by Nick Arpaio, former postdoctoral fellow in the lab, Clarissa Campbell, and Peter McKinney, and Clarissa Campbell and Stas Dickey showed that extrathymic differentiation of regulatory T cells dependent on intronic enhancer CNS1 can be promoted by the metabolic output of commensal microbial community. It has been well demonstrated by Kenya Honda, Chi She, and many others that antigens encoded by commensal microbial community represent major antigenic stimuli that promote extrathymic differentiation of regulatory T cells, with their primary site being in the gastrointestinal tract, where commensal microbiota represents the largest source of non-self antigenic material and plentiful ligands for innate immune sensors. So what Clarissa and Nick were able to show that certain classes, two major classes of microbial metabolites, short-chain fatty acids, in particular butyrate, and particular types of secondary bile acids, and similar observations were made by others, Wendy Garrett, Hiroshi Ono, Dennis Kasper, Dan Littman's lab, that microbial metabolites would promote extrathymic differentiation of regulatory T cells. And once these cells are generated, then Stas and Clarissa were able to demonstrate that by a feedback, this generated extrathymic regulatory T cells would promote establishment of beneficial microbial community that would support metabolic function of the intestine and its microbiota under physiologic conditions. So now I would like to discuss recent work by Saskia Kemmers in the lab, and go back to the thymus and discuss what you would call developmental niche for regulatory T cell differentiation, considering that that regulatory T cells are potent modulators of different types of the immune responses. The question in our mind was whether their differentiation is scaled by the availability of key niche factor, and we believe that key niche factor for their differentiation is interleukin-2, as we have already discussed. So with this in mind, Saskia set out to look for the cellular source of interleukin-2 in the thymus, and work for multiple laboratories indicated multiple potential sources of interleukin-2, ranging from dendritic cells to thymocytes, various types of thymocytes, gamma delta T cells, NKT cells, alpha beta T cells, as was shown by Michael Farrar recently, and even epithelial cells were suggested to produce interleukin-2 during thymic differentiation of regulatory T cells. So, well, first question that Saskia meant to ask, whether there is a requirement for continuous interleukin-2 production for thymic regulatory T cell differentiation. And this question was important because mice deficient in interleukin-2 or interleukin-2 receptor subunits develop early autoimmune manifestations, lymphoproliferative syndrome, become anemic, and very seriously sick. So it's requirement for continuous IL-2 production for thymic regulatory T cell differentiation has not been really established. So with this, Saskia undertook, decided to inducibly ablate conditional allele of interleukin-2 with tamoxifen-inducible Cre recombinase expressed in a ubiquitous manner, and found that upon inducible ablation in mice, there is a phenotype, there is a decrease in FOXP3 positive, CD25 positive regulatory T cells in the thymus, and this decrease was similar to the one that was observed in mice with germline deficiency in interleukin-2, although mice at that time were by and large clinically healthy. So at the next step, Saskia undertook fate mapping, genetic fate mapping of cells with the history of interleukin-2 expression using interleukin-2 endogenous interleukin-2 locus encoding Cre recombinase, and this allele was combined with a ROSA26 recombination reporter allele, where TD tomato fluorescent protein reporter was, and its coding sequence was preceded by a stop cassette flanked by LOXP sites. Upon Cre recombination, stop cassette is excised and tomato reporter is expressed. And that is shown here, where Saskia was able to identify small but clearly identifiable subset of cells with the current and historic expression of interleukin-2. She also found that the vast majority of these cells express, the vast majority of the cells that had history of IL-2 expression were CD4 single positive thymocytes, with very few CD8 single positive thymocytes showing history of expression, and not any other cells. And that this slide shows quantification of percent of cells among T cell receptor positive cells. And this approach also enabled tracking of cells with a history of interleukin-2 expression, not only in the thymus and the periphery, over time. And this is shown in lower slides. So then, of course, then Saskia attempted to confirm production of interleukin-2 by stimulating different subsets of isolated thymocytes ex vivo. But that experiment really was not satisfactory, because it really doesn't show ability, the production. It shows the potential to produce interleukin-2. With this in mind, she then turned to the data sets that were generated in the lab by Stas Dickey, where Stas assessed chromatin accessibility in different subsets of thymocytes that were isolated and divided into some immature CD4 single positive thymocytes, mature FOXP3 negative, CD25 negative, single positive thymocytes, CD25 positive FOXP3 negative subset that we already discussed, that is highly enriched for precursors of regulatory T cells, and FOXP3 positive thymocytes. And Saskia decided just simply to look at accessibility of interleukin-2 locus in the different subsets of cells, and she found that interleukin-2 locus is accessible only in CD25 positive FOXP3 negative thymocytes, whereas in other cells, in other subsets of cells, it was not. That indicates that the cells within this population that is highly enriched in precursors also highly enriched in cells that are poised to express interleukin-2 gene. And these cells, consistent with previous work, experience stronger T cell receptor signal, as shown here by the level of expression of TCR reporter that was generated in the lab some time ago by Young. It's similar to reporter that was generated previously by Chris Hochquist. It's NOR77 back transgene that encodes a red fluorescent protein, and you can see that both CD25 high and CD25 intermediate low population of FOXP3 positive cells experienced strong T cell receptor signal based on the reporter expression, but also the CD25 positive FOXP3 negative cells experience strong T cell receptor signal, and this plot just quantifies this expression. And then using genetic approach and ablation of conditional interleukin-2 allele, early in thymocyte differentiation using proximal promoter LCK driven Cre transgene, Saskia was able to show that thymocyte production of interleukin-2 is important for generation of regulatory T cells, and that was not, whereas ablation of interleukin-2 in dendritic cells was inconsequential, and so was ablation of interleukin-2 in peripheral T cells using distal LCK promoter. And this experiment excludes possibility that peripheral differentiated effector T cells re-entering the thymus might serve as a source of interleukin-2 for extrathymic differentiation of regulatory T cells. Now, this is the summary of a number of studies that Saskia performed using in vitro modeling of interleukin-2 production in response to T cell receptor engagement by thymocytes, in vivo modeling of interleukin production, and single cell RNA-seq analysis of interleukin-2 producing of cells producing interleukin-2 led to the following conclusion that mature CD4 single positive thymocytes that experience T cell receptor signal upregulate CD25, and this T cell receptor signal that the cells experience in some cells would result in production of interleukin-2. So the cells frequently would express CD25, and interleukin-2 will be produced either in paracrine or autocrine manner. And many of this, a number of the cells likely undergo apoptosis, as we observed apoptotic gene signature in cells with interleukin-2 expression. A few of them can become FOXP3 positive cells, and some of them are likely going out in the periphery where they continue to potentially produce interleukin-2 for the maintenance of peripheral regulatory T cells. So together, this data suggested that self-reactive CD4 single positive thymocytes serve as a key source of interleukin-2 and therefore function as a niche cells during regulatory T cell differentiation, and regulatory T cell generation in the thymus is scaled by self-reactivity, namely by production of interleukin-2 by self-reactive thymocytes. So together, work from many labs and work from our laboratory suggested that there is this intimate connection between FOXP3 gene and interleukin-2 signaling. And interleukin-2 signaling plays an essential role really in biology of regulatory T cells. So then the question that we had was how such a new IL-2-centered regulatory T cell functionality would emerge during evolution where FOXP3 gene would suddenly appear. And the question that we thought to address in this study is whether there is a non-redundant role for an ancestral FOXP family member in regulatory T cells. FOXP family member, to which FOXP3 belongs to, has four genes that emerged upon 2 rounds of whole genome duplication during transition from invertebrates to vertebrates. FOXP3 is the latest arrival with other family members which share a number of features, including conserved forkhead DNA binding domain and coiled coil motif that is required for dimerization. They are capable of both homo and heterodimerization, and a number of labs showed this, including work by Deepai and Rudra, former postdoctoral fellow in the lab, who showed that FOXP3 can heterodimerize with FOXP1 and FOXP4. And FOXP1 is more abundantly expressed in T cells and regulatory T cells, and FOXP4 is expressed at the lower level. So what Kasia Konopatsky, a former graduate student in the lab, and Yuri Pritikin, a very talented postdoctoral fellow in Christina Leslie's lab, computational biologist at SKI, decided to ask. They decided to ask whether FOXP1 and FOXP3 bind to shared or distinct sites within the genome of regulatory T cells, whether FOXP1 deficiency affect binding of FOXP3 through chromatin, and whether there are non-redundant role for FOXP1 in regulatory T cells. And by doing quantitative chip sequencing for FOXP1 and FOXP3 in cells that contain FOXP1 and lack FOXP1, conditional allele of FOXP1 was generated many years ago by Yuri Rubtsov, former postdoctoral fellow in the lab. They found that essentially FOXP1 binding sites are essentially fully overlapped. So with relaxed threshold, with relaxed stringency, only 54 sites are not bound by FOXP3 among the sites that are bound by FOXP1. And there are some FOXP3 bound sites that are not bound by FOXP3. The four-cat motif enriched at the sites was indistinguishable, and so was its motif. What was surprising to observe that not only at the sites where FOXP1 and FOXP3 bind together, but also at the sites where FOXP3 binds singly, Kasia and Yuri observed that FOXP3 binding was diminished. Genome-wide FOXP3 occupancy in regulatory T cells was diminished in the absence of FOXP1, and that is best reflected by this box plots shown here, where when the binding was assessed at peak flanks, that is chromatin chip sequencing peaks, or in all accessible sites, that was no difference between a signal from FOXP1 deficient and sufficient cells. But for both FOXP3, all FOXP3 peaks, FOXP3 overlapping peaks, and FOXP3 only peaks, there was a decreased binding of FOXP3 in cells lacking FOXP1. Although we don't know yet the explanation for it, we are thinking of potential redistribution of FOXP3 in the absence of FOXP1 to multiple sites, because in the absence, since the binding is similar, then in the absence of FOXP1 would reduce the overall amount of FOXP containing proteins to bind the sites. So, but perhaps more importantly for this talk and for the point that I would like to make is that FOXP1 deficient regulatory T cells were functionally impaired, and that was shown by cell transfer into mice lacking regulatory T cells jointly with effector T cells in breast cancer model, where this tumors transplanted in orthotopically into mammary fat pads were growing more slowly in mice where regulatory T cells were devoid of FOXP1. And we believe that to a significant degree, impairment in functionality can be traced to diminished expression of CD25 in FOXP1 deficient regulatory T cells, and that is shown here. CD25 was diminished in FOXP1 deficient expression of in FOXP1 deficient regulatory T cells, different tissues, and phosphostat-5 phosphorylation in response to titrated amount of interleukin-2 was also diminished. So what that suggests that that the emergence of regulatory T cells may potentially be helped through cooperativity of the new member of transcription factor, FOXP transcription factor family FOXP3, and its ancestor FOXP1, with important role for FOXP1, essential and non-redundant role in cell intrinsic control of interleukin-2 receptor expression and interleukin-2 responsiveness in regulatory T cells. So not only interleukin-2 signaling is required for extrathymic for both thymic and extrathymic differentiation of regulatory T cells, Takatoshi Chinen and the lab, through ablation of essential components of interleukin-2 signaling pathway, being interleukin-2 receptor beta chain, alpha chain, or downstream transcription STAT5 showed that interleukin-2 receptor signaling is essential for the function, suppressor function of regulatory T cells, and the loss of any of these components resulted in severe and fatal autoimmune and inflammatory lesions similar to those observed in mice lacking regulatory T cells. Similarly, second signal, T cell receptor signal in differentiated regulatory T cells is required for their suppressor function and differentiation of naive regulatory T cells into effector cells. And that was shown by former MD PhD student in the lab, Andrew Levine, by inducible ablation of a conditional T cell receptor allele in differentiated regulatory T cells in tamoxifen-inducible manner, or by flipping T cell receptor repertoire displayed by regulatory T cells to an unrelated and unselected repertoire. And all this manipulations, genetic manipulations, resulted in a loss of suppressor function by regulatory T cells. However, in addition to suppressor function that is T cell receptor dependent in regulatory T cells, work by Nick Arpaia and Jesse Green in the lab suggested that there is innate-like non-immune function of regulatory T cell that is T cell receptor independent, and this function lies in the ability of regulatory T cells to support tissue maintenance. Both work by Nick and Jesse showed that regulatory T cells, when subjected to ablation of T cell receptor, that results in the loss of their suppressor activity, were nevertheless were perfectly capable of producing EGF family member amphiregulin and support tissue repair function. And the cue to production of amphiregulin by regulatory T cells was provided by alarmin, interleukin-33, released upon necrotic cell death, and pro-inflammatory cytokine, interleukin-18, and a number of other labs, including Diane Mattis, Christophe Benoit, and many others, focused on this connection between interleukin-33 signaling and amphiregulin production. So before I finish, I would like to quickly draw your attention to the finding that we and others have made now a long time ago that was visibly paradoxic observation that activated regulatory T cells sometimes co-express their lineage specification factor, FOXP3, and transcription factors that define effector T cell response, different effector response types, such as TBET, GATA3, ROR gamma T, and this co-expression of FOXP3 with other transcription factors that can promote either tissue accommodations, adaptations, and tissue responses, or immune effector responses have been noted by multiple different labs listed with some work, but not all the work cited on the right side. So one can reason that upon exposure to pro-inflammatory cytokines, regulatory T cells acquire some of the expression of these transcription factors. And the question that Andrew Levine asked several years ago was whether expression of this transcription factor represents a reversible activation or stable differentiation state, and what might be biological function of this expression, especially if it's a reflection of stable differentiated state of regulatory T cells. So with this, Andrew focused on regulatory T cells that express transcription factor TBET for one simple reason, because expression of FOXP3 was unaffected by TBET expression in regulatory T cells, and FOXP3 expression does not affect expression of TBET in regulatory T cells. They can be essentially uncoupled genetically. So, and to address this question, Andrew generated fate mapping alleles, inducible reporter allele for gene encoding transcription factor TBET, TBX21, red fluorescent protein CRER, and decided to undertake genetic fate mapping of the type that I described to you were differentiated when regulatory T cells, when exposed to tamoxifen, can now express fluorescent reporter, now yellow fluorescent protein, and can be mapped, and cells with unstable and stable expression can be readily identified. So in this study, Andrew found that Tibet regulatory T cells are continuously generated at steady state in response to self-environmental antigens. And he found that upon transitional unstable Tibet expression in Tibet low expressors, regulatory T cells acquire stable Tibet expression and maintain its expression, not only in permissive conditions and type 1 cytokine-rich milieu, but also under non-permissive conditions, for example, in settings of warm infection. And he also observed de novo generation of Tibet-expressing regulatory T cells from Tibet-negative regulatory T cells during bacterial and viral infection. So that raised the question what the function of the cells might be. And similar to other reports, Andrew found that Tibet expression in regulatory T cells had at best limited role in control of Th1 responses and was dispensable for ability of the cells to control autoimmunity. But I think that the question that Andrew had that at the time was whether it's not Tibet, but other genes that are covariantly expressed with Tibet and regulatory T cells define the functionality of Tibet expressing regulatory T cells. So decided to reverse the experiment, not to ablate Tibet and regulatory T cells, but to ablate FOXP3 in Tibet expressing regulatory T cells. And this showed that mice subjected to ablation of FOXP3 in Tibet expressing regulatory T cells that result, as I told you, in the loss of identity and functionality of the cells resulted in severe type 1 dominated autoimmunity. And similarly, more complicated mice that he generated where he was able to selectively ablate Tibet expressing regulatory T cells using diphtheria toxin resulted in similar type 1 dominated autoimmunity with type 2 and type 3 responses, Th17 responses unaffected. Andrew was further able to perform a gain of function experiment using complicated alleles that he generated, and these genetic alleles are schematically shown on this slide, where in this mice, Andrew was able to selectively deplete regulatory T cells lacking Tibet expression and to leave only Tibet expressing regulatory T cells. So upon provision of tamoxifen, cells that express Tibet were lost expression of diphtheria toxin receptor on the cell surface, and therefore were spared from diphtheria toxin mediated ablation, where cells that lack expression of Tibet were continued to express diphtheria toxin receptor and were subjected to ablation. So in this study, Andrew was able to show that Tibet expressing regulatory T cells were selectively poised to prevent type 1 inflammatory responses, but failed to efficiently control type 2 and type 3 inflammatory responses. And how we explain this, and a work by Alejandro Mendoza in the lab, who collaborated with Andrew in the studies, showed that Tibet expressing regulatory T cells commingle, if you will, with Th1 cells and CD8 T cells, the cells whose responses they control. Alejandro found that the cells colocalize and are effectively interacting with their target cells, and this is likely mediated by their guidance cues provided by chemokine receptors, integrins, and other GPCRs. So together, this study suggested that there is essential heterogeneity of regulatory T cells that can be generated upon, in this case, their diverse inflammatory exposures that may enable formation of spatially distributed regulated units of the adaptive immune response that are comprised by pro-inflammatory cells, amplifiers, anti-inflammatory cells, regulatory T cells or decelerators. And in this way, these units may resemble neuronal circuits. So I would like to finish by acknowledging people who did the work, and there are a number of current lab members whose names I brought up. Mikhail helped, is a bioinformatics specialist in the lab who helped with a lot of single cell analysis. And these are former members of the lab whose work I also quoted, and Yuri is a postdoctoral fellow. He's about to start his own lab at Princeton very soon in the lab of my close colleague and collaborator, Christina Leslie, at the Computational Systems Biology program at Sloan Kettering Institute. Thank you for your attention.
Thank you so much. That was a phenomenal talk. Thank you very much for your efforts to prepare it. That I learned a lot. Thank you.
Thank you so much.
So I will now share my screen. And I hope you can see it. And basically, just reminding everyone that as a format for the Global IMMUNOTALKs, we have questions and answers via Twitter. And so Dr. Rudensky had agreed to answer the questions. And so this will be, for that, you need to first search for the Global IMMUNOTALK account. Then you need to find the tweet that says ask questions for Dr. Alexander Rudensky here. That tweet is already in the account. And then you reply to that tweet with your question and mentioning the hashtag Global Immuno. And so the questions will be answered with the account Immuno Speaker. So again, thank you so much for a terrific talk and for teaching us much more about Tregs and of your amazing work. We really appreciate it.
Yes, thanks a lot.
Thank you very much. Thank you for listening.
With that, we will close, and just reminding you that our next Global Immune speaker is Dr. Ron Germain from NIH, and we hope you all join us again next week. Bye, everyone. Thank you.
Bye-bye. Thank you.